So this video will walk you through just the basic information for the PTC lab in case you're absent or in case you just want to kind of refresh what we did during the day. So really it's a lab to see if you have a gene called the bitter tasting gene, which just allows you to taste things that are bitter, that bitter flavor. So what is PTC? What are we really talking about? So the way that your brain kind of recognizes different tastes has to do with different cells. So your tongue has different, your taste buds basically, they're just different cells that have different types of receptors that recognize different molecules. So for example, if you eat something sweet, that sugar molecule, when it binds to those receptors on the sweet cells, it sends a nerve impulse to your brain that says you're eating something sweet because something has stimulated those sweet cells. That sugar molecule is not going to bind to those receptors on the bitter cells or the sour cells because it doesn't recognize it. It's not a match to those cells. So it's only going to match to those sweet cells. Okay. So that's basically how it works. And it just stimulates or sends a signal to your brain that says you're eating something sweet. So these are just some examples of the genes for these different, that code for these different proteins for these cells. So, here's a sweet cell that is going to recognize that sucrose. Glutamate is the umami taste, bitter, salty, sour. So you can just see it's different proteins that are folded differently for these cell receptors, and they're going to bond to different molecules or substances that are going to trigger that signal. So this PTC, which actually stands for phenylthiocarbamide, it was actually accidentally discovered, this whole genetic taste ability. In the 1930s, Arthur Fox, he was a scientist, he synthesized some phenylthiocarbamide, or PTC, and some dust actually escaped into the air. And so his lab mate, could taste that dust in the air and said, oh man, it tastes really bitter. But Fox didn't taste anything, and he thought that was really strange. Why is this person tasting this bitterness, but I don't taste anything at all? And so that just kind of led to many other studies with taste, and especially using this PTC substance and how it could trigger that bitter tasting gene, basically. And so later studies showed that this inability to taste PTC is recessive. And so now we know there's really about 30 different genes that all kind of code for these bitter tasting receptors. So all these different there's obviously lots of different substances that have a bitter taste. And so you can have, there's genes that code for those cell receptor proteins for all these different substances. So something to keep in mind is just because if you're not a taster of PTC, that doesn't necessarily mean that you're not a taster of other bitter substances, right? Like you could still taste something else that tastes bitter. It just means that specifically you don't have the gene for this specific bitter substance. And so the PTC receptor gene is called TAS2R38. So like I said, all these numbers and codes is just because there's lots of different tasting genes, some for sweet, sour, salty, bitter, and there's lots, at least about 30 or so bitter tasting genes. So lots of different taste genes. The one we're looking at is this one right here. And you can see it's highlighting here let me circle it for you. I guess you can kind of see it in red, but the mutation that occurs, so it's really a non-taster is a G here, and then the taster is a C. And so if I had just something to keep in mind for later when we start analyzing our results, remember you have 2 copies of every gene, right? One from mom, one from dad. And so if you're a non-taster, that means that you really have 2 of these, GCACT, right? One from mom and one from dad. And so both of these would have the G and the G, right? Non-taster. If you're a homozygous, let me go through. That means you have this C here, right? Because you have a mutated copy from mom and from dad. Or this is, I don't actually know which is the original, which one we're technically calling mutated. I guess we're really probably calling this the non-taster, the recessive copy. But either way, it's just this point mutation, this difference in this gene. So if I'm let me change my color here. If I'm a heterozygous, which means I have a capital T and a little t, right? That means that, I'm going to have all of this beginning and end is going to be the same, but I'm going to have the C, just like I did here, for my tasting gene. And then for my non-taster gene, I'm going to have the G. Okay, so you'll have, I guess the non-mutated and one mutated, if you want to think about it that way. So just something to think about when we get into our analysis results, right? You could be heterozygous, which means you have one of these, of the C change, that point change. So the purpose of this lab, right, is just to determine your genotype for the PTC tasting gene, whether you're homozygous recessive, homozygous dominant, or heterozygous, okay? And so by doing this, or in order to determine this, we're going to isolate your DNA from your cheek cells. We'll amplify it using PCR, and then we'll analyze it using gel electrophoresis, okay? So I've broken down the procedure for each kind of step that we're going to be doing. So in this video, I'm not going to necessarily go over every single step. I'll just kind of in general tell you what we're going to do, because you don't really need every step of the procedure if you're not in class. So this is kind of the basic thing that we'll do. So this is really going to be day 1. Right here. So we'll isolate your DNA. We're just going to use this should be a 0.9 solution, percent saline solution. And you're just going to switch it around and then spit it out into a cup. And then we'll take some of that liquid from the cup. We will do a couple things to it to kind of isolate that DNA from your cheek cells, and then we'll add it into the thermal cycler, so that in order to do the PCR steps. So like I said, there's a couple things that are going to happen in between there, and I'll post the slides on Schoology so you can see all the steps, but that's what we'll be doing on day 1. On day 2, we're actually going to do this right here. We're going to digest your DNA. So that means that we're going to be using that restriction enzyme that will cut. It's actually going to cut right at this section here. I think it's right here in between these 2 C's, but I'll have to double check. I think it's right here in between those 2 C's. I'll scroll down. I think I have that in the slides somewhere. So we'll add the restriction enzyme. And then you have to incubate that for a little while longer. There's lots of incubation steps, which is why we have to kind of separate this up. But on day 2, we're also going to make the gel. So we'll have to make an agarose solution with the comb in it, to make the wells. And then that has to set kind of for a little bit too. So there's a couple steps in there that we'll do. And then day 3, we will actually run the gel and analyze your sample. And so we're going to compare an undigested, which means that we don't have a restriction enzyme, with your digested. And so what that's going to do is this undigested is going to kind of serve as your control. So if you don't see anything in your undigested column, that basically means it didn't work. Something happened. Maybe you didn't have enough DNA. Maybe, I don't know, just there could be multiple reasons why, but that it just means it didn't work. So that's kind of like our control for this. Okay, so each person will do their own undigested. That way, because you should have done everything the same to both samples. So let's see if there's anything else that I want to go over before. These are just, like I said, all the little steps, like broken down for when we're there. Yep. Okay. So here's our restriction enzyme. So let me change my color here. So it's going to recognize this GGCC, and it's going to actually cut right in the middle of it, in the middle of the GGCC. So over here, we don't have a GGCC sequence at all. There's a GGGC, but there's not this right here. And so we can see now, basically our restriction enzyme has just cut right in the middle of that sample. Okay, so when you look at your gel electrophoresis, right, this is really what the undigested sample will look like. It's just going to have this whole gene, right? This whole section here of your, and it's longer than this. This is just these couple of 10 base pairs, but it's really 220 base pairs long is the gene. So you'll have that whole section, and you should see just one band. If you're a homozygous taster, right? Remember we've cut, you have these, we're cutting it at this point where over here we have 44 base pairs. We have 176 base pairs over here. So we're going to actually see those two. Let's see, the 44 comes down here, and then this one's right here. So we'll see those two bands there. So one thing to think about that you'll need to discuss possibly in your analysis section of your lab report is, what do you think a heterozygous person will look like here, right? What will they look like on the gel if they have one copy of the taster gene and one copy of the non-taster gene right there? Okay, so that's just something to think about. And I believe that is all that I'm going to go over in this video. There will be some other videos posted later on with some results. So if you are absent for some reason and you don't get to analyze your own sample, then you'll just discuss results of another sample that I'll give you the results of, probably using like a video or a picture or something like that. So you'll still be able to analyze some DNA and kind of discuss the different, the possible genotypes based on the results, but it just may not be your own DNA. So look out for those videos to come later.